there, this is your instructor Christina Howard, and I am recording for the first portion of the Endocrine System lecture for Biology 242 at Clark College. So with that, here we go. I'm going to pause here and take my screen off so I can write on it as I talk to you. So if you see the screen go blank for a second, that's what's happening. Oops, there we go. All right, and away we go. So let's move on. So the first thing we have to discuss before we get into the nitty-gritty of the endocrine system itself is intercellular communication in general, or at large. So the primary mode of communication between cells that we are going to discuss in this presentation is this one, endocrine communication, which occurs via blood vessels. So hormones get dumped into blood vessels, which is what this guy is, by cells. And I want to stress that this is primarily via the process of exocytosis. And so anything that travels in the blood as a chemical mediator we call a hormone. So that's kind of the primary focus of this particular chapter of your book. It doesn't mean that other kinds of communication don't exist. They absolutely do. It's just that since this is the endocrine chapter, this is going to be our primary focus. So we also have direct communication. So this is via gap junctions, which remember are arrays of protein that poke through a membrane and allow a cell to share its contents with its neighbor. We've also got paracrine communication. So this is when a cell releases something into the extracellular fluid, and instead of going into the blood, it simply diffuses to nearby cells to provide them with a signal. So these are fairly limited as far as how far they can broadcast, but they are very common and very effective. So an example of this will be something that we discuss a lot more later on, which is interferons. Interferons, if you're curious, are chemicals that cells use to tell their neighbors that they have a viral infection and to take antiviral measures. So if you're burning with curiosity about that because of COVID-19, stay tuned. We will talk about that when we get to the immune system and lymphatic system. And then down here at the bottom, we have a kind of communication that you guys are very familiar with after taking 241, and that is, of course, synaptic communication. So this is a chemical synapse. And then this one is actually also considered a synapse, so this is an electrical synapse. But again, we are on the endocrine chapter, so the endocrine communication mode is our primary focus. Alrighty, so moving right along. Basics of endocrinology. So endocrinology is the study of the endocrine system. It's also a specialty in medicine. So if you want to be something other than a general practitioner and you want to be a specialist, this is one of the areas that you can go into. So hormones are secreted into the extracellular fluid, and they travel a very, very short distance before they eventually enter the blood. So we functionally consider hormones to enter the blood directly, even though that's not quite true. It's just that the time that hormones spend in the ECF is so limited that we just call it endocrine, even though it resembles paracrine a little bit. So blood circulates hormones throughout the body, and that's because blood vessels go nearly everywhere. Notable exception, cartilage for one thing, that's avascular, and other avascular tissues, but most of the tissues of your body are quite vascular, which means that if blood goes there, hormones can go there too. The name for the cells that respond to hormones we call target cells. And another way to say this, and this is on a hormone by hormone basis, mind you, so almost all of the cells in your body are target cells for some hormone. It's just which one is it, which one or ones. So the word target cell is used on a hormone by hormone basis. Like, is this cell a target cell for growth hormone? If yes, it will express receptors for growth hormone. If no, it won't. So this is a very specific piece of nomenclature. Receptors are interesting because a cell can change how sensitive it is to a given hormone by increasing or decreasing the number of receptors it is currently expressing. So that's what accounts for this range between 2,000 and 10,000 receptors per cell, and that's per cell per hormone. So not just total receptors, but for one single hormone. So if a cell is sensitive to, say, 8 to 10 hormones, that number can get big really fast, right? So one of the things we're going to do is talk about how exactly hormones affect the cells that express receptors for them. So these bottom two images are starting to get at that. So we've got a transmembrane protein receptor bound to something called a G protein, which you just started to learn about at the end of last term. And then we have this new idea here, which is a steroid hormone just skipping the plasma membrane altogether and going straight into the nucleus. So we'll discuss both of those things as we move forward. One thing that we need to do first is discuss the chemical classes of hormones. So the reason for this is what chemical properties a hormone has is going to determine how it can act and how it can travel in the blood. So amino acid derivatives and peptide hormones, oops, I don't want to use my PowerPoint marker because it is error prone, sorry. So amino acid derivatives and peptide hormones, these are polar molecules, which means that they are water soluble, which means that they travel very easily in the blood without any disruption. Lipid derivatives, not so. So you know from Biology 160 that water and fat don't mix, right? So lipid derivatives, and I'm going to put an asterisk on here, and thyroid hormone, T3 and T4 is the abbreviation for that. Lipid derivatives like prostaglandin and thyroid hormones need help being in the blood. So lipid derivatives are obviously fat-based. Excuse me, pardon my yawn. Meaning that they're not miscible in water, and therefore they need to have some kind of a molecular taxi or carrier to allow them to make their way through the bloodstream to the tissue that needs to interact with them. Alrighty. So here is a sort of summary slide showing you the different kinds of hormones. So we've got amino acid derivatives over here. And these are what they sound like. So they are hormones that are made out of amino acids by modifying them chemically. Peptide hormones, which are synthesized in the same manner as proteins are. And then lipid derivatives. So I'm going to pause here and ask you a sort of idea linkage question. What organelle or organelles do you think are responsible for a lot of these lipid-derived hormones? So I invite you to press pause here and think about the answer for a moment, and then see what I say once you come back. So interestingly, and I will show you the pathway for this when we get to our recitation session, but I'm not going to do it in lecture, the smooth endoplasmic reticulum, unsurprisingly, is involved, and that's because, remember, lipids are produced by the smooth ER, but also mitochondria have an important role specifically in producing these. So let's write this down, SER. And mitochondria. All right, so those are the two organelles that are primarily responsible for making lipid-derived hormones. So we're going to go through these piece by piece. So amino acid derivatives are exactly what they sound like. They're amino acids that you get in your diet, and then your body takes those amino acids and runs them through a series of chemical reactions that structurally change the molecule to go from being just kind of a regular old garden variety amino acid to being a hormone. So here we go. We've got two precursors. Hi there. Sorry about that. We got cut off because the software I was using, I didn't realize had a 10-minute limit on it. So I've switched to another piece of software that I am not test driving, and now we should be good to go for a longer duration since I paid for this. Seems the key is if you pay for the software, they don't cut you off at 10 minutes. Alrighty, so we were talking about amino acid derivatives and how the body just simply takes amino acids that you eat in your food, in familiar foods, for example, turkey, and then does some fancy chemistry action to them and turns them into useful signaling molecules. So tyrosine is one amino acid, and sources of tyrosine generally include animal products, so meat, dairy, and fish. And this is the precursor or substrate to make thyroid hormones and also catecholamines, which you're familiar with by now. These are epinephrine and norepinephrine, which I'm circling here, but also dopamine. So dopamine is also a catecholamine. Perhaps more familiar to you is tryptophan, and that's because there's been numerous pop science news articles written about how tryptophan is found in turkey, and that's the reason you get sleepy after you eat turkey at Thanksgiving, for example. So this is kind of a more common knowledge one. So tryptophan is going to be used to make melatonin, and melatonin is a pineal hormone, and it's involved in circadian rhythms. So sleep-wake cycles, day-nighttime stuff. We'll get to more on that later. Oops. All right, peptide hormones. So peptides refers to a peptide bond, which remember is the special bond between amino acids. Which I'm going to abbreviate AAs. So we have two classes, and these are glycoproteins. So a glycoprotein is a long chain of amino acids, which I'm illustrating here in blue, and then there are sugars hanging off of the glycoprotein. So these are greater than 200 amino acids long, and they have carbohydrate side chains. So these include thyroid stimulating hormone, not the same thing as thyroid hormone, luteinizing hormone, and other anterior pituitary hormones. The other class of peptide hormones is short peptides or small proteins. So these are pretty little. They're less than 200 amino acids, sometimes as small as 9, but also as large as 198. And these are a much larger group, which means they're also more diverse. So, finally, we get to the lipid derivatives. And one thing that's important to note here is that they are lipid derivatives, meaning that these are going to be constructed. Excuse me. Pardon. For some reason, recording makes me really sleepy. Not sure why. They're going to be constructed from dietary fats. So we have two classes. We will talk about eicosanoids a little bit more when we get to the immune system, because eicosanoids are important signaling factors used by white blood cells. So leukotrienes allow triangulation of leukocyte activity. That's why they're named that. And prostaglandins are involved with inflammatory responses, so responses to infection and tissue damage. Steroids are derivatives of cholesterol. Yes, cholesterol, that molecule that everybody tells you is bad all the time. It's not actually bad. In fact, if you do not have enough cholesterol, or the building blocks thereof, you cannot make the sex steroid hormones, which is what these are. And sex steroid hormones are important for things like reproduction, obviously, but also sex drive and maintenance of secondary sex characteristics. So skin and hair, for example, as well as external genitalia and breast tissue. So if you don't have enough cholesterol on deck, those processes get halted. I would be remiss if I did not also address the fact that stress hormones that come from the adrenal gland are also considered to be steroid hormones, as is calcitriol, which is the most active form of vitamin D. Vitamin D is fat-based and therefore is also considered to be a steroid hormone. We'll talk about all of those things at least a little bit. We've already talked about vitamin D about as much as we're going to. We did that last term in the integumentary system. So one of the things that I mentioned previously was that the chemistry of different hormone classes determines how they can travel in the blood. And that's absolutely true. So some of them freely circulate, and that is because they are water soluble. So as I mentioned previously, the water soluble ones are the polar ones, and these are the peptide hormones and the catecholamines. Some are transported by carrier proteins in the blood. So these tend to be lipid soluble and or be thyroid hormones. So you can't mix oil with water. Similarly, you can't mix fat. So you can think of the transport of steroid hormones and thyroid hormones as these guys require a little molecular taxi or Uber or whatever you prefer. Regardless, they need a ride to get to where they're going. They can't just go there by themselves. And that's related to their solubility. So, with regard to freely circulating hormones, meaning ones that are not bound to a carrier, ones that can travel in the bloodstream by themselves, how long they're in the blood really varies, and it depends on the specific physiological context as well as which hormone it is. So that time can be anywhere from a couple of minutes to an entire hour. Again, and I'm going to put an asterisk on here. The asterisk here means it really just depends. So I'm not going to ask you to be any more specific than connecting this time frame to this group of hormones and then knowing that they are water soluble. So I'm helping you connect those three dots. So what happens to them? Well, one of three things. They diffuse out and bind to their receptors. They are metabolized and excreted by your liver and your kidney. And this often happens after the receptor step as well. So they can either be metabolized before they ever hit a cell with receptors for them, or once they have bound, done their job, and hop off the receptor, they can be metabolized and excreted by the liver and the kidney at that time. Some enzymatic breakdown will occur in the plasma or interstitial fluid, but only if the appropriate enzymes are there. So only if enzymes for those hormones exist. For example, one that you already are aware of is the one that breaks down catecholamines, and that is catechol O-methyltransferase, which breaks down epinephrine, norepinephrine, and dopamine if those hormones escape from the synapse they're in. And one thing that's missing here is I want to add little receptors for all of these hormones, because really that's what's happening here is these hormones are binding to receptors on the surface of these cells. Okay, so hormones bound to carrier proteins. These are analogous to a taxi or Uber or Lyft or what have you, like I said, or a lazy river if you prefer. So a lazy river is a water park ride where it literally is just a river that moves real slow. And most people that are in the lazy river are sitting on floaties. So they're being carried in this waterway by floaties. The only time they're really not on the floaties is if they are entering or exiting the water. And so what that means is if you took a snapshot of the lazy river at any given time, most people are going to be attached to a flotation device. A very small number of them will be off of the flotation device. So it's kind of the same idea as the hormones that are bound to carrier proteins. So the majority of the ones that require a carrier are attached to their carriers. Only about 1% are free. So again, these are thyroid hormones and steroid hormones. So we're using thyroid hormones in these two pictures as an example, but that doesn't mean they're the only ones that behave this way. It's just the ones that the illustrators picked to draw. That's all. And then this is released from carrier proteins as the free portion is used, meaning that as T3 and T4 hops off to go do its job, its spot is replaced. What this allows you to do is pretty powerful, and that is the hormones themselves are not active as long as they are bound to their carrier, which means that you can keep several weeks' supply actively circulating in your bloodstream before you start to run out. So as long as you're actively replenishing your supply and there are sufficient thyroglobulins or other carrier proteins, you have a backup supply of that hormone that you can easily mobilize just by having it disengage from its carrier and enter the appropriate tissue. So it's a good way to store an activatable pool of hormone rather than having to synthesize new hormone every single time you want to use it. Okay, so this next segment is going to resemble some of the nervous system stuff that we did last term pretty closely. So we're going to talk about how exactly hormones act on their target cells. And I want to preface this, like with everything, the exact mechanism for how the hormones do this is really going to depend on the hormone and, for example, which G protein is activated and what exactly that does. So what enzymes is it activating? What ion channels is it opening? That is determined on a hormone by hormone basis. So we're not going to get any more specific than that in general. Excuse me. Because we can't. If this were an endocrinology class, I would absolutely expect that of you, but it's not a reasonable thing to expect of you at this level. So it's going to seem like I'm being vague. I'm doing that on purpose. I'm sparing you from extra information that you don't need at this stage in your path. So the general goal of hormones is to change the metabolism of their target cells somehow. And if we know that enzymes do most of the work of the cell, what that means is that changing enzyme activity is going to likewise change cell metabolism in a way that is favorable to homeostasis. So the whole point of all of this is to make sure that homeostasis is maintained. So in order to affect change on cells, they can open or close ion channels. They can also activate genes by landing on promoter regions of genes. So they can cause the synthesis of enzymes or structural proteins. And they can increase or decrease the synthesis rate of proteins by either stimulating or suppressing transcription, which, remember, from 160 is DNA to RNA, and then mRNA translation, which is messenger RNA to proteins. So they can change how fast or slow a particular enzyme or protein is transcribed and translated. So we have two overarching methods for how a hormone acts. One of them is more similar or more familiar to most of you than the other one, and that is the hormone binds to a receptor. So a receptor is a protein that's sitting in the membrane, and it has a special spot on its surface that is only the right shape for the hormone that it wants to bind, period. In this case, though, the hormone binding to the receptor, as you can see, doesn't open up anything. So it's not the case that this receptor is a channel. Instead, the shape change that results from the protein receptor binding to its substrate, the hormone, turns on an internal protein, a G protein, and that ends up causing an increase in cyclic adenosine monophosphate. So cyclic AMP is an important signaling molecule. It's considered to be a second messenger, and it does all kinds of stuff that's very dependent on what cell you're looking at. The alternate outcome is that the hormone can pass through the membrane without issue and bind to a receptor either in the cytoplasm or in the nucleus of the cell. So I'm going to take a moment here and ask you, what chemical class of hormone do you think would bind in a cytoplasm or in the nucleus rather than on the surface of a cell? So why don't you pause the video now and take a moment to think about it. Okay, so hopefully you had a moment to think about it. The correct answer to my question is lipid-based. So anything that can get through a membrane with relative ease without having to be transported is going to be passing through the outside of the cell and acting on the inner portion of it. Okay, so which hormone classes do what? Well, as you might expect, since peptide hormones, catecholamines, and interestingly, eicosanoids are amphipathic. These are going to bind to receptors in the cell membrane and activate a G protein. So they're not going to enter. Notice that this hormone is not going into here. That's not happening. But rather, when it sits on top, that activates changes inside the cell. So G protein activation results in an increase in cyclic AMP and generally an increase in calcium ions. So both of these things go up. So if the particular enzyme being acted upon is adenylate cyclase, that means that we're going to convert ATP to cyclic AMP. And if the G protein is going to activate PDE, which is, here, I'll write that over here actually. Phosphodiesterase, then we're going to convert cyclic AMP back to AMP, which reduces enzyme activity. So increase in AMP really means an increase in enzyme activity, and a decrease means a decrease in enzyme activity. So cyclic AMP can go up or down. So finally, phospholipase C, which is what PLC stands for, and DAG, diglycerol, these are outcomes of G protein activation that can both open calcium channels, so letting in calcium from the extracellular fluid, or releasing it from a storage area like the endoplasmic reticulum. It really just depends. In that case, calcium will then bind to calmodulin, and it complexed with calmodulin is what allows it to act as a second messenger. So this is a summary slide of all the stuff I just told you, but now we've zoomed out a little bit. Oh, let me get rid of all that stuff. And I can also show you the outcomes down here in the form of some examples. So these are some first messenger examples for each kind. So what does first messenger mean? First messenger means the hormone. The second messenger is cyclic AMP, or this, adenosine monophosphate, or calcium. So the first messenger is the thing that initiates the process inside the cell. The second messenger is whatever thing is turned on or off inside the cell. Okay, so amplification is the first case. And I know I just explained this, but repetition is our friend when we're learning new concepts, so I want to make sure that I repeat things and be explicitly clear about them. So amplification is powerful because what it means is, because you can go from one hormone to many thousands of cyclic AMP for one binding event of one hormone, that means that a relatively low amount of water-soluble hormones is capable of having a dramatically large effect. And this is because they initiate what is called a receptor cascade. So that is what you're seeing here. This process is the receptor cascade. So it's just the series of events that amplify a signal after a hormone binds to its receptor. That's all. So hormone activates G proteins. G proteins activate adenylate cyclase. Adenylate cyclase increases cyclic AMP. Cyclic AMP activates protein kinases. You can think of kinases as enzyme activators. And enzymes perform chemical reactions. So adenylate cyclase produces thousands, like I said, of second messengers, meaning that this signal goes from one hormone to a 1,000 magnitude response really quickly. So what does cyclic adenosine monophosphate do exactly? Well, it can either activate or inhibit enzymes depending on whether it's activating phosphodiesterase or adenylate cyclase. And as I showed you via calcium, it can also open ion channels. So phospholipase C is the primary opener of the ion channels, and primarily as well, excuse me, those ion channels are calcium channels. So the calcium comes from the endoplasmic reticulum, specifically smooth, or the extracellular fluid, so the fluid that's outside of cells. Once calcium activates calmodulin, which is a protein that binds to calcium, and as the name suggests, modulates it, and calmodulin is going to activate enzymes. Steroid hormones work a little differently. And remember, that's because they don't have any trouble getting through a membrane whatsoever. So steroid hormones have a really easy time accomplishing the very tricky task of getting into a cell. And that's because they are lipid soluble. So they have no issue crossing through the plasma membrane because they can get through that hydrophobic zone in the middle of the phospholipid bilayer. So these are going to tend to bind to either cytoplasmic or nuclear, and then therefore DNA-based receptors. And these alter cell activity and structure primarily by altering gene expression, but not always. But that's enough information for you guys for right now. So, thyroid hormone. These in general increase ATP production, and that shouldn't surprise anybody because most people are familiar with the idea that hyperthyroidism, of which there are several kinds, but generally, if your thyroid is overactive, you're going to be burning more ATP than usual, and that means a decrease in weight. The opposite is also true. So hypothyroid means an increase in weight, because hypothyroid means that thyroid hormone is not stimulating metabolism to the same extent that it was when you were at rest previously. So obviously these are going to happen via altering cell activity. So altering how cells use fuel and things like appetite, etc. So those are all going to be sort of influencing factors of thyroid hormone's effect on cells and tissues. Okay, so let's summarize here. Hormones' effect on target cells. We know that in general, they're capable of changing enzyme activity either in the positive direction, so increasing it, or in the negative direction, decreasing it. They're also capable of opening and closing ion channels, and I want to stress that not always, but often, usually calcium channels. And then they also activate genes, so they can cause the synthesis of new enzymes, or the downregulation of enzymes. And they can also cause the synthesis of structural proteins as well. So they can increase or decrease the synthesis rate of proteins by stimulating gene transcription and mRNA translation, just like in the previous kind. All right. So, one of the things that's going to be different for 242 is we're going to do some activities, and these activities are going to be for points. So during the normal term of Biology 242, what we do is we do this blood tracing exam where you have to pretend you're a little blood cell and you have to take me on a walk through the vasculature piece by piece. Mark and I have decided not to do that this term, but I did want to provide you with a points cushion, so I'm designing these case study activities that will be worth 10 points each and that you'll turn in electronically on Canvas, and those will make up for the loss of the 40 points from the blood tracing activity. I think you'll find that this activity is more applicable to your careers and also easier than doing anything else. So that's what we're going to do. So there's 4 of them. They're worth 10 points each. And assuming you show me you make a good faith effort and attempt to think critically, there's no reason you shouldn't get full credit for those assignments. So that's kind of your buffer, is those 40 points. All right, and then that's all I'm going to do for today. I'm finding that I have an easier time uploading my videos if I break them into shorter chunks. So thank you for your attention, and I will see you in the next recording.